Hello friends, hope all are fine. Welcome to my channel Sa Biology Plus. Today we will come to know that what compound microscope is, its different parts and their functions. But before that, will you please consider subscribing my YouTube channel? Your subscription and viewership are often a lot to me. First of all, let us know what compound microscope is. A microscope that uses multiple lenses for higher magnification is known as compound microscope. As already said in previous video, a microscope has two major characteristics. Number one, magnifying power, and number two, resolving power. When the magnifying power increases, the image size also increases in direct proportion. Resolving power, on the other hand, is the ability to distinguish between two closed space points. So when the resolving power increases, the image becomes clearer, sharper and more detailed. There are different types of compound microscopes such as bright field microscope, dark field microscope, inverted microscope, fluorescence microscope and phase contrast microscope. This figure illustrates the different parts of a compound microscope. The lowermost part of the microscope is a metallic base on which the whole microscope rests. Connected to the base, there are a pair of vertical elevations known as pillars. The body of the microscope is joined with these pillars by means of an inclination joint. By loosening this joint, the body of the microscope can be bent to a suitable inclined position for convenient viewing. Another part of the microscope is the curved arm. It is a curved structure connected to these pillars. The structure held body tube, coarse and fine adjustment knob and the stakes. The body tube, which is a cylindrical structure, is associated with a revolving nose piece at the bottom. This is the nose piece which can be rotated. It is associated with several objective lenses of different magnifying power and resolving power. The coarse adjustment knob is used to move the body tube up and down through a long distance so as to bring the objective lens into focus. On the other hand, fine adjustment knob is used to bring the objective into sharp focus by moving the body tube up and down through a small distance. The upper part of the body tube, which is comparatively narrow, is known as draw tube. Into the upper part of the draw tube, eyepiece is inserted for examination. One of the most important parts of a microscope is the source of light. In old model microscope, a mirror which is held to the base is the source of light. When light falls on the mirror, it gets reflected, which is then allowed to pass through the condenser, which is positioned above. The condenser forms a cone of light. In modern microscope, the source of light is inbuilt at the base. It can be halogen or LED lamp. Whereas in case of fluorescence microscope, arc lamp is used for illumination. When all light is allowed to pass through the condenser, then the specimen gets brilliantly illuminated. As a result, it cannot be visualized properly. So it is necessary to control the amount of light that passes through the specimen. This is achieved by using diaphragm. The main function of the diaphragm is to regulate light intensity. It manages image contrast, brightness and field depth. There are four types of diaphragms. Number one, iris diaphragm, which is functionally similar to the iris of our eye. Iris is located right below the substage condenser. This is the substage condenser which forms a cone of light that illuminates the specimen. Iris is usually attached to the bottom of the condenser lens. The diaphragm is a metal structure consisting of several small overlapping metal leaves that can be adjusted to form a perfectly circular aperture through which light will pass into the condenser, which then forms a cone of light in order to illuminate the specimen. When the diaphragm is opened, more amount of light reaches the specimen. As a result, magnification and resolution increases, but field depth and contrast decreases. And when the diaphragm is closed, less amount of light reaches the specimen. As a result, contrast and field depth increases, but resolution decreases. The diaphragm is open particularly when viewing under high magnification. Number two, field diaphragm. In advanced microscope, a second diaphragm which is located right above the source of light is used. This is known as field diaphragm, whose main function is to control the overall size of illuminated area. It reduces stray light and glare without affecting the quality of image. Number three, disc diaphragm, which is used in school level microscope. It is a disc-shaped structure containing several fixed holes of different sizes, and the disc can be rotated to change the aperture. Number four, annular diaphragm, which is used in phase contrast microscope for viewing living unstained specimen. In inverted microscope, the condenser is located above the stage, pointing downward and directing light down through the specimen. In such type of microscope, objectives are located below the stage. There are different types of substage condensers. Number one, Abbe condenser, which produces a solid cone of light that illuminates the specimen. Such type of condenser is used in standard bright field microscope. Number two, dark field condenser, which by using a opaque top prevent direct light from entering the objective lens, thereby creating a dark background. Dark field condenser produces a hollow cone of light. The direct oblique light rays from all sides to strike the specimen. This enables direct observation of living, unstained and transparent specimen such as microorganisms which will not be visible in bright field. Number three, phase contrast condenser, which uses an annular diaphragm to produce phase contrast. This condenser also produces a hollow cone of light. In addition, in phase contrast microscope, a phase plate is present behind the objective lens. This phase plate shifts the phase of direct light by lambda by 4, attenuating its amplitude. Such type of microscope is therefore useful for examining living and unstained specimen. Iris, which is located below the condenser lens, can be opened or closed by using a handle which is attached to it. The condenser lens assembly can be moved up and down by using the condenser knob. This knob is used to optimize illumination of the sample by focusing light on the specimen. It also enhances image brightness, resolution and contrast. In upright microscope, above the condenser, there is a horizontal platform known as the stage on which the specimen slide is placed. There are two types of stages. One is simple stage, which is found in simple microscope. On the other hand, in advanced microscope, there is mechanical stage on which the specimen slide can be moved smoothly along X and Y axis by using two adjustment knobs. The main function of the condenser is to focus a cone of light onto the specimen. As the light from the condenser passes through the specimen, it is differentially absorbed, refracted or scattered. This creates contrast. As a result, we can see different structures within the specimen. So when the light passes through the specimen, it is diverged and then enters into the objective lens, which is placed above. In upright microscope, at the bottom of the body tube, there is a revolving nose piece. It is associated with three objectives which have different magnifying power and numerical aperture. The main function of the objective lens is to gather light that comes out through the specimen. This lens forms the real magnified and inverted image, which is then further magnified by the eyepiece, which produces the final upright magnified image. So friends, this is all about different parts of a compound microscope and their functions. For more updates, subscribe my YouTube channel, Biology Class.